Good morning. It's September 10th, and this is your daily brief in science. Here's everything you need to know. A recent study has unveiled a startling trend in urban rainfall patterns, revealing how cities are reshaping weather dynamics globally. Published in the Proceedings of the National Academy of Sciences, the research analyzed rainfall data from 1,056 cities worldwide between 2001 and 2020. The findings indicate that over 60% of these cities experience more precipitation than their rural counterparts, a phenomenon termed urban wet islands. This urban precipitation anomaly is largely attributed to the urban heat island effect, where cities are warmer than surrounding areas. This temperature difference can lead to concentrated rainfall, akin to a sponge being squeezed, which heightens the risk of flash flooding in densely populated regions. The implications of this trend are significant, as it raises serious concerns about urban flooding, especially as extreme weather events become more frequent. The study emphasizes the urgent need for infrastructure adaptations, such as the implementation of sponge cities, designed to absorb excess moisture and reduce flooding risks. Current weather models often overlook the urban influences on rainfall, highlighting a gap that city planners must address in future urban development strategies. The research, led by Devna Yogi from the University of Texas at Austin, marks a significant advancement in understanding urban climate dynamics and the role cities play in altering regional weather patterns. As urban areas continue to affect climate through temperature changes, terrain alterations, and pollution, the global wet island phenomenon has intensified over the past 2 decades. This combination of increased rainfall and impervious surfaces poses a heightened risk for flash flooding, underscoring the need for innovative flood preparation strategies. Carbapenem-resistant Klebsiella pneumoniae, or CRKP, is emerging as a significant threat in healthcare settings, particularly as a leading cause of hospital-acquired lung infections. The Centers for Disease Control and Prevention has classified CRKP as an urgent public health threat, emphasizing the need for alternative treatment options beyond traditional antibiotics. Hospitalization due to bacterial pneumonia and sepsis remains a pressing concern, with high rates of mortality associated with these infections. In a recent study published in Science Advances, researchers Pankaj Baral and Prabhu Raj Joshi are exploring the complex interactions between the nervous and immune systems during CRKP infections. Their findings indicate that lung-innervating nociceptor sensory neurons, which are generally protective, may actually worsen the severity of pneumonic sepsis in the context of CRKP lung infections. This unexpected role of nociceptor neurons raises questions about their function during infection and potential implications for treatment strategies. The researchers suggest that targeting these nociceptor neurons or inhibiting their signaling pathways could lead to the development of non-antibiotic therapies. To advance this research, the team is working with the University of Kansas to screen small molecules that may serve as inhibitors of nociceptor signaling. Additionally, researchers at Kansas State University have noted that immune response dysfunction contributes to the complications associated with these infections. Funding for this critical research initiative is provided by several organizations, including the National Institutes of Health and the American Lung Association, highlighting the collaborative effort to address the challenges posed by CRKP. A recent study from the University of Copenhagen has unveiled the significant role of the OSER1 protein in promoting longevity. This research opens up new avenues for potential treatments aimed at addressing the challenges of aging. Understanding the mechanisms of OSER1 in both cellular and animal models is crucial for tackling age-related diseases and improving overall human health. The findings indicate that insights gained from these animal models can be relevant to human aging processes, enhancing our understanding of longevity. The discovery of OSER1 presents promising new drug targets for various age-related conditions, including metabolic, cardiovascular, and neurodegenerative diseases. Published on August 21, 2024 in Nature Communications, the study details how OSER1 reduces oxidative stress and extends lifespan across multiple species. The article, titled FOXO-regulated OSER1 reduces oxidative stress and extends lifespan in multiple species, underscores the broader implications of OSER1 in longevity research. These findings shed light on human longevity and suggest potential interventions for health issues associated with aging. Researchers have unveiled a groundbreaking molecular engineering technique that offers precise control over organoid development using customizable DNA microbeads. This innovative approach allows these microbeads to transport various signaling molecules specifically tailored for different tissue types, enhancing the functionality of organoids. The DNA microbeads are not only customizable in their mechanical properties but also scalable for production and biocompatible with organoid cultures. Developed by an interdisciplinary team from Heidelberg University, the Max Planck Institute for Medical Research, and the Cluster of Excellence 3D Matter Made to Order, this technique addresses limitations associated with traditional external morphogen supplementation. The study shows that internal morphogen gradients can effectively guide organoid development. In experiments, DNA microbeads loaded with a WNT agonist significantly increased the diversity of cell types in retinal organoids, closely mimicking an in vivo retinal environment. For the first time, researchers successfully induced retinal pigment epithelial cells to form adjacent to neural retinal tissue through localized release of signaling molecules. The technique was tested on retinal organoids derived from the Japanese ricefish Medica, known for their rapid development compared to other organoid models. Experts suggest that this advancement could accelerate research in human development and disease, as well as enhance organoid-based drug research. The microbeads can release growth factors or signaling molecules at specific times and locations within the organoid, resulting in more complex and realistic tissue structures. The research findings were published in the journal Nature Nanotechnology, marking a significant step forward in organoid technology. A recent study published in the Journal of Cell Science has unveiled significant insights into the FEM1B gene and its role in the cell cycle, shedding light on cancer cell proliferation. The research delves into a process known as stop codon read-through, where the protein synthesis machinery occasionally bypasses the stop codon, resulting in the creation of longer proteins. This genetic alteration may help explain the heightened susceptibility of humans to cancer compared to other primates. Evolutionary analysis indicates that the stop codon read-through phenomenon is unique to humans and chimpanzees, potentially stemming from a single nucleotide insertion that occurred around 10 million years ago. Sandeep Eswarappa, an associate professor at the Indian Institute of Science, emphasizes that such read-through can significantly impact a protein's localization, stability, and function. The FEM1B protein is crucial for marking other proteins for degradation, thereby regulating various cellular processes, including the cell cycle. Researchers found that stop codon read-through produces a longer, more unstable version of FEM1B, which paradoxically leads to its degradation and reduced protein levels. By utilizing CRISPR-Cas9 technology, the team eliminated the sequence responsible for read-through in cancer cell lines, resulting in increased FEM1B levels, enhanced degradation of target proteins, and a delayed cell cycle, ultimately reducing cancer cell proliferation. In mouse models, cancer cell lines that lacked read-through demonstrated slower tumor growth. The research team is now focused on uncovering the molecular mechanisms behind stop codon read-through, aiming to develop targeted therapeutic strategies for controlling tumor progression. Additionally, analysis of data from human cancer patients revealed that higher expression levels of the FEM1B gene correlate with better survival rates, reinforcing the study's findings. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.